Hello, everyone. Welcome back to my video tutorial for spatial data analysis. So today I'm going to show you how to recreate the same Seurat cell clusters from the Xenium Onboard analysis. So the dataset I'm going to use for demonstration is the human breast cancer replicate one dataset. Previously, I used this dataset to show you how to create a Seurat object and analyze the data using Seurat pipeline. You can use this link to download the data. Once you download the data and also unzip the data, you will have a folder named as out. So here it contains all the files to analyze this dataset. You can see the files. We have the analysis folder. Today we are going to use the data from this folder. And also we have the cell feature matrix and the cell feature matrix H5 file. We have the cell boundaries, CSV cells. We have the nuclear boundaries, transcripts, and the other files are the data. You can use the Xenium Explorer to analyze the data. If you want to understand the data in each files, you can go to the Xenium website. It has the details for each data file. In the file folder, you can see the analysis summary. You can see this file is a HTML file. If you click this file, it will open the analysis summary in Internet Explorer. Okay, you can see we opened here. Here has the summary, decoding, cell segmentation. We want to see the analysis data. You can see down here. On the left-hand side are the cell clusters on the image, and on the right-hand side are the cell clusters. You can see from the Xenium Onboard analysis, there are 20 cell clusters in total in this dataset. So today we aim to create the same cell clusters as the Xenium Onboard analysis. So now we can go to R to create a Seurat object first. We can use the node Xenium function to create a Xenium object. Let's run this code to create a Xenium object. Okay, you can see we created a Seurat object named the Xenium object. Let's click the Seurat object and have a look at the data. You can see in this object we have 313 genes. In total, there are 167,780 cells. At the moment, we have the cells that doesn't show any gene expression. So we can use the subset function to remove the cells that didn't show any gene expression during the data collection. So let's remove the cells with the 0 count. So if we have a look at the object again, you can see now we only have 166,363 cells. So next we can normalize the data. Then later you can identify, for example, differentially expressed genes. Let's normalize the data first. So you can also scale the data and run PCA, but we are aiming to recreate the same Umap as the Xenium Onboard analysis. So we don't need to run scale the data and run PCA. So now we need the data information from the analysis folder. You can see in the analysis folder, we can click the analysis folder. You can see we have the clustering information, differentially expressed genes, PCA information, t-SNE, and Umap. So we want to create the same Umap as the online analysis, we can go to the Umap folder. You can see here are the gene expression 2 components. Let's click. We have a CSV file here. Let's open this file. So you can see we have the barcode in the A column, Umap1 information in B column, and the Umap2 information in the C column. So we can load this Umap information into R. So first we can use read CSV function to load the Umap information. Okay, you can see we have the Umap. You can see it also has 166,363 cells. It is the same number of the Seurat object. That's the reason you need to remove the zero count. Sometimes if you open the data set, they actually perform the analysis without removing the zero count. So you need to compare the barcode with the Umap and the Seurat object to decide if you need to run the steps to remove cells with the zero counts. So let's have a look at the Seurat object again. You can see at the moment for the reduction it's 0. So we don't have any PCA or Umap information. So now we need to change the Umap information as a matrix data. You can see now it is a large matrix. Then we can use the create dim reduction object function to add the Umap information into the object. Let's run. If we have a look at the Seurat object now, you can see now we have the reduction, the Umap information for the Seurat object. So we have the Umap information. Next, we need to add the cell canisters. I showed you we have 20 cell canisters in total from the onboard analysis. We can go to the onboard analysis again. You can see here we have 20 cell canisters in total from the onboard analysis. Here is the graph-based cell canisters. If you click the arrow, you can see there are canisters for gene expressed k-means k equals 2 until k equals 10. For example, if we click the gene expression k-means k equals 10, then we only have 10 cell canisters. You can see on the left-hand side, the image was changed to 10 cell canisters. So now we need the cell canister information from the analysis folder. Let's go to the cell canisters. You can see the canistering. We have the graph-based canister k-means 2 to k-means 10 canisters. So I'm going to show you how to recreate the 20 cell canisters. Use the gene expression graph canister information in here. If you want to create the other canisters, you can follow the same step. But I'm going to use this information for the demonstration. We can open the CSV file for the canisters. You can see the A column is the barcode, B column is the canister information for each cell. So now we can load the canister information into R. Again, we are going to use the read CSV function. Let's load the cell canister information. Now you can see we have the canisters. In total, we have the same number, 166,363 cells. So before we add the canister information in the metadata, we can have a look at the metadata. You can see in the metadata, we have the origin, ident, uncount, and features. And also we have other information here, but we don't have the cell canister information. So now we can get the canister information. We know it is in the cluster data frame, then add it into the Seurat object as a Xenium cluster. Let's add the cluster information to the Seurat object. Now we can have a look at the metadata again. You can see now in the last column, we have the new column named the Xenium cluster. We have the cluster information. So now we have the Umap and also cluster information. We can just use the node to look at the cell clusters. You can see we have the reduction of the Umap. Then we group the cells by the Xenium cluster information. Let's make the clusters. Okay, you can see we generated the Umap. We have 20 cell clusters. If we compare our cell clusters with the onboard analysis, you can see we have the same shape, same number of cell clusters, but the color looks different because the onboard analysis uses different color schemes. So now we can use the image node function to look at the cell clusters on the tissue. We can run. Okay, you can see we generated the spatial image. We can zoom in. You can see we generated the image. It is the same image as the onboard analysis. Again, we are using different color code to label the cell clusters. If you want, you can change the color for each cell cluster. Okay, that's today's video tutorial. I showed you how to recreate the Seurat cell cluster and the spatial image from the Xenium Onboard analysis. Once you recreate the Seurat object with the same cell cluster, you can perform further analysis such as the identification of differentially expressed genes. So I'm going to stop from here for today's demonstration. I hope my video tutorial could help your data analysis.